and letting folks know to refresh their pages. And it looks like we are good to go. Hi everyone, my name is Emily and I'm the Community Manager of 2 Diabetes here working with Diabetes Hands Foundation. And we have a super exciting live interview lined up for today, which I know we have a slew of people in the chat room already excited to hear all about the work of ViaCyte Incorporated. Today we're meeting with Paul Laikind, President and CEO of ViaCyte Inc., and Lisa Hepner, the founder and creator and producer of Vox Pop Films. And I'm going to have each of them introduce themselves to you rather than telling you about them while they're sitting here watching me do so. Paul, let's start with you. And please start with just a little bit about who you are and what you do at ViaCyte.
Okay, well, thank you. I'm happy to be here to talk about the exciting work we're doing there. So, as you said, I'm Paul Laikind. I'm the president and CEO of ViaCyte. I've been with the company for about 4 years. The company's been around a lot longer than that, but I joined them at the time when they were starting to move towards the clinical evaluation of the product we're going to talk about today. My background, Ph.D. in biochemistry, so coming out of the science side. I have been what's often referred to as a serial entrepreneur in the biotechnology industry, having started a series of companies, taken them public, and had them acquired. But I think ViaCyte is by far the most exciting company I've worked with to date.
Fantastic. And Lisa, please introduce yourself and tell us a little bit about who you are and what you do with Vox Pop Films.
Hi, thanks so much for having me on, Emily. My name is Lisa Hepner, and I run a company in Los Angeles called Vox Pop Films with my husband, Guy Mossman. And I think our most exciting project to date is the human trial, a documentary feature that is following ViaCyte in their quest to cure type 1. And it's been a thrill, actually, to go behind the scenes of the medical research that ViaCyte is doing. It's been very eye-opening. I should add, I have a personal interest in this. I've had type 1 for 25 years.
Woohoo! It couldn't get more personal than that as a personal interest in what ViaCyte is doing. So with that, Paul, what is ViaCyte doing? We're all incredibly excited about the company. For those who have heard the name but don't know why we're so excited, please give us a breakdown of what you're working on.
Okay. So ViaCyte is a company. We are what's called a regenerative medicine company. We're focused primarily on a cell replacement therapies. What our big focus, and the reason we're talking today, and for over 10 years this company has been focused on really transforming the way type 1 diabetes is treated, essentially providing a functional cure. And the way we're doing that is we're developing a product that we call a stem cell-derived islet replacement therapy. So I think this audience probably knows what islets are. I don't have to explain that. But I think one way of looking at it is, about 100 years ago, insulin was discovered, or insulin was injectable insulin that came onto the scene, and it really changed diabetes from a fatal disease to a chronic disease. But for the last 100 years, we've really just kind of improved on that a little bit. We've made insulin better. We've made ways of injecting insulin and pumps. We've gotten better at monitoring glucose and all. But at the end of the day, we're still doing what we did 100 years ago. We're monitoring blood glucose. We're monitoring diets. We're monitoring exercise. And then we're injecting insulin to control the disease. And as everybody here knows, it's an imperfect answer. It's certainly a major impact on quality of life, but it also has a big effect on the longevity. It has effects on long-term complications. So what we're trying to do at ViaCyte is really change that whole equation and go in and do what the body has lost its ability to do, which is to produce that insulin naturally from the beta cells. And not only that, to replace the other cells in the endocrine system that also help to control that blood glucose and metabolism. And so we do that by starting with an embryonic stem cell line that we isolated many years ago. We then differentiate that to a pancreatic progenitor cell, so it's a cell that is essentially destined to go on to become a islet, to become beta cells and alpha cells and delta cells that produce insulin, glucagon, and somatostatin. And then we go from there. We encapsulate that in a device because we need to protect these cells from the immune system. And Lisa, you might want to mute, by the way.
Yeah, let me hit my mute here. Sorry.
Okay. So we then encapsulate that, and then we implant that under the skin, and if everything goes as planned, what we've seen in the animal studies is these cells will become islets under the skin and control the blood glucose just as a natural beta cell will. So that's it in a gist. I'd be happy to take other questions about that.
Nope, that's it. We're pretty much done.
Okay. Well, we'll see you later. It was great seeing you.
Thanks for coming, everyone.
Oh.
So I want to get into a little bit more about the specifics of the product that ViaCyte is working on. It's called VC01. And Paul, can you talk about exactly, it's a two-part process. What are those two parts and how exactly is it intended to work?
Sure. So there's really two, what in the industry or in the field, what we call platform technologies. The first technology is being able to take a stem cell, in our case an embryonic stem cell, and for those of you who may not be familiar with that, stem cells are cells in the body that can go on to produce any other cell in the body. Okay, so they're what's called pluripotent cells. They can differentiate and become any other cell in the body. So we take this stem cell starting material. We have essentially a bank of these cells, both master and working cell banks. And when we're ready to manufacture our product, we take cells from that, we expand them, and then we go through a directed differentiation that takes them from that embryonic stem cell up to a pancreatic progenitor cell. And what I just said in about 20 seconds took about 8 years of work, okay, to develop, to be able to do that, to go from that embryonic stem cell up to the pancreatic cell, and do it in a reproducible, scalable, and regulatory compliant way. So now we've got a population of cells that are destined to go on to become islets, and that's great, but what do we do with them? Okay, so we need to get them into the patient. These are human cells, but they're not the patient cells, and therefore those cells would be essentially attacked by the immune system if we were just to put them in, and so we need to protect them from that. And so that's the second part of the technology, the second platform, if you will. And we have developed a macroencapsulation device, and you can think of that like a tea bag, okay? And we place these cells into that device, and then they are implanted under the skin. After implantation, at least in the animal models, they differentiate up to the islet cells and then produce produce insulin in response to the glucose levels of the patient. So it has potential in a mouse model. One of the interesting aspects in a mouse model is we've seen, when we put these human cells in a mouse model, we take the normal blood glucose level of a mouse and change it to the human level. So we essentially humanize the glucose metabolism of that mouse for the life of the mouse. So it's very effective there, and we can show if we completely destroy the beta cells, just like you do have with autoimmune disease, destroying beta cells in type 1 diabetics, we can continue to control that blood glucose without any issue. So it has potential to be an extremely effective therapy for patients that would, again, take them now from that chronic disease to a disease that you no longer have to think about, and that's our goal. To get this to the point where we're controlling that disease at such a level that patients no longer have to even think about it. I know from I don't have type 1 diabetes in my family. I'm happy to say that. Okay, but I know many people who have type 1 diabetes. And one thing I'm told all the time by patients is this is a disease you never get to quit thinking about. And so, I think with this product, we change that.
So far, there's been a lot of research done in mice, right? And I was looking on the ViaCyte website, and I see that the preclinical disease models are what we call those mouse trials. Is that correct, Paul?
Yes, that's correct.
Okay. And at this point now, humans, you're at the point where humans get to be involved in some clinical work, which is very exciting because as somebody has already noted in our chat room, mice are different from humans. And we've heard so many times about really, really exciting diabetes-related breakthrough in mice, which at this point I think a lot of folks are a little jaded about hearing about how many different ways we know how to cure mice with diabetes. And we still can't seem to do it in people. Can you talk a little bit about now what is this next piece that ViaCyte has started? And then Lisa, I want to move over to you and how much of this you've been able to witness so far.
Yeah, so I think people are absolutely correct to be jaded about mouse models. They are a surrogate for us. They allow us to test certain things. But the key is being able to translate what we've seen to a mouse into a patient. The mouse is actually, interestingly, an imperfect model for us because when we're treating patients, we're putting human cells in humans. When we're treating a mouse, we're putting human cells in a completely different species. The immune response to those cells is very different when you're in the animal model. So it really is not the best of models for doing this, but it's all we have. And insulin, happily, is insulin, whether it's human insulin works in a mouse just as it does in a human. So it does give us a lot of information, but the key is then to move to the point where we can translate this and move it into the patients. And that's where we've been for over a year now. We filed an IND, which is an investigational new drug application with the FDA to get clearance to go ahead and start testing. And we began the clinical trials about a year ago. The trial itself is a 2-cohort trial, this first trial. The first cohort is a subtherapeutic dosing of cells, so we give a small dose of cells. It's not meant to be therapeutic. It's allow us to look primarily at safety and tolerance of the product, but also to develop the techniques to implant it. And we know, even from the animal models, when you do cell therapy, when you first put these cells in, if you think about it for a second, when we have these cells prior to implantation, they're living in an incubator at a nice constant 37 degrees. They've got oxygen being bubbled through them. They've got all kinds of nutrients and everything. So it's a really nice environment for a cell, if you will. We then take that cell, we put it in a device, and we drop it into a war zone, okay, because basically we've done a surgical incision to put it under the skin. The body is responding to that. The body is responding to the fact that a foreign body has been put under the skin. So these cells are now in essentially a war zone. And so what we've been really focused on over the last year is developing the techniques to minimize the amount of cell loss during that transplantation. So there's enough cells there to essentially, you go through a period where you see that cell loss, but get to a point where then the cells can proliferate and fill the device and then give you the effectiveness you want. So that's what all the focus has been on now. Once we're convinced that that is working and that we've optimized that 100%, or at least close to 100%, then we would move on to a second cohort of patients where we give a fully therapeutic dose, and then we will look for efficacy, insulin production, glucose regulation, hypoglycemic reductions.
So that, as we noted, is incredibly exciting stuff. Now, Lisa, as a person with type 1 who's documenting this whole process, there's a lot that I wonder about your experience of this. But the first thing I want to ask you is, how did you choose ViaCyte as the object of the subject of of the documentary that you're creating, which, as you said earlier, is called The Human Trial. And are you able to unmute yourself? There we go.
The reason I'm muting myself is in downtown LA, it sounds like Armageddon. I don't know what's happening right now. The mayor might be coming through. Sorry, I apologize. Should I come back? Let me just hear if this is clearing. Okay. Can you guys hear me okay?
Yeah.
Crazy. It's really crazy out here. So why did we choose ViaCyte? So I don't know, Emily, if you remember, I was on your show, like I guess 2 years ago, 2013, when our film was called Patient 13. And at that point, we were following another team of researchers. And what I realized in the process of filming this team of researchers is that not everyone gets to the clinic. Not everyone gets to test their device or their protocol on humans. It's actually not easy to get there for many different reasons. So we knew that this project was going to be on hold. The story that we've been following, it was going to be on hold for a while. So we actually went to JDRF and we spoke to some of the people in the science, like Peter Lemedico, people who deal with industry, and we said, you know, so who is close to a clinical trial? Who's close to getting into humans? Who is also doing encapsulation? And you know, from JDRF to also we spoke to the International Diabetes Federation, we did our own research, we came to ViaCyte, and thankfully they're in our backyard. They're in La Jolla, or rather San Diego, we're in LA. And it was, and Paul can attest to this, it was, you know, it took us some time to establish a relationship and to establish access. Because as you can imagine, shooting behind the scenes of a major clinical trial that could cure a major chronic disease, it really hasn't been done before. There have been documentaries that have gone behind the scenes of orphan diseases, which is great and cool. And there was a wonderful documentary that was nominated for an Oscar called The Life According to Sam. It was on HBO. But there really hasn't been sort of unfettered access to going behind the scenes of a clinical trial in progress. So we're breaking new ground, and Paul and his team are breaking new ground, and by Paul agreeing to sit with us and say, hey, okay, we'll let your cameras in with a few conditions, it's been an extraordinary opportunity. So we were very fortunate. We got the access after, you know, some negotiation, and then when we started to film was actually when ViaCyte got their IND approved by the FDA, which meant they could proceed to the clinic. So we were actually in the room when ViaCyte spread the news to their team. Paul knew what the answer was, but the rest of the team didn't. So you might see that in our trailer and in our teaser that's on our website. So we've been privy to some pretty good moments, and you know, just recently there's been some news about the trial, you know, that Paul's talking about, and we were there filming Paul explaining that to the staff. And it's those kind of moments that are so real and authentic that if you were to tell in the past tense, it would be intriguing, but it wouldn't be as dramatic. So it's worth every amount of effort we've made, and obviously, you know, Paul, they give us, it's not easy for them to always allow us in, but they've been really accommodating. And maybe I'll let Paul speak to why he's doing it. But I think we all see the power in getting the message out to the largest population possible. You know, we know there is such an unmet need with people who want their story told around diabetes, specifically type 1, that I think this audience will be a large one and an important one. And I just want to add one other thing. Paul is talking about the embryonic stem cell research that is so critical to the success of their product. I mean, I want to point out how pioneering their trial is. It's only the 3rd ever in the United States and the 6th ever in the world. So, you know, there are a lot of eyes on this trial, just even outside the type 1 community. And we're just lucky to be behind the scenes of it.
Now, what she didn't mention is she also filmed us bowling and various other things.
Well, you got it. I mean, you know, we need to also humanize the scientists behind the research. You know, people are more than just white coats. You know, you've been in this for a long time, Paul. You've been in the field for like over 30 years. And you've said to us on numerous occasions that you're the most passionate about this particular project. It has the most potential to affect millions of people.
Absolutely.
So you know, I think when we film you guys bowling or going to Indian Princess Camp with your daughter, or you know, seeing you guys out at your luau, your company picnic, it just shows that you're people first, and that your passion to cure this disease has been powerful since, like, what, all the iterations of the company. It's very rare to have a biotech company stick to one product.
Yeah, no, actually, it's astounded me when as I said, I joined 4 years ago. We have people in the company who have been working towards this project for as many as 15 years. And, you know, we have employees in the company, and to be honest, this company has, you know, we've been funded, but we've gotten a lot of funding from JDRF, from the California Institute of Regenerative Medicine, which is fantastic, but it's not the same as having a big fat bank account. And, you know, so we've kind of made ends meet and continued forward and pushed this forward into the clinic. And, you know, to have this team stick with it, and the dedication that they have had to really seeing this through and finding a cure for type 1 diabetes is inspiring. And it certainly inspired me when I decided to join the company.
So, yeah, go ahead. Sorry, Emily.
That's okay. So in terms of that inspiration, I think it's really interesting to talk exactly as you're saying about the people who are involved, the actual people, humans who are involved in this effort. And it's true that folks who work on cures or treatments, that they're actual people who are actually dedicated to that effort. And sometimes the patients can lose sight of that a little bit, because we usually don't have access to the folks who are actually making the products that we use. So that's part of why being able to interview both of you is so exciting. But I wonder, Paul, if you could talk a little bit about sort of the emotional side of the pursuit that you're involved in for you, the person who doesn't have type 1 diabetes or an immediate family member with diabetes, but still is very dedicated to this effort.
Yeah, well, I mean, I am, you know, I trained as a scientist, and I started out as a research scientist, pretty much basic research, but focused more on the medical field. And so I'd say that is what's motivated me my whole career, is the excitement, the adventure of exploring new areas and all. And, you know, to be able to have the impact we're having, or what we hope to have, on a disease that is as widespread and as difficult as this one, you know, that's tremendously motivating to me. You know, as Lisa mentioned, I've said a number of times, you know, I've been working in this industry for 30 years. I've worked on a lot of different projects and product opportunities, and I don't think there's ever been one that I found as exciting as this one. It's not often in this industry that you get an opportunity to bring out a product that doesn't just incrementally impact a disease, but really changes it fundamentally. And that's what I think this product has the potential to do. And I just want to say this, you know, I've been here 4 years. As I said, the team at ViaCyte has been working on this for much longer. So as excited and as dedicated as I am, that's just the tip of the iceberg. The rest of that team is even way more, gets way more credit than I do for what's happened here and moving this forward.
And I think that's really, oops, there we go. I think that's really apparent in some of the video footage that I've already seen, that Lisa has put together, is that you're really showing how emotional this pursuit is for the folks at ViaCyte, and how personal it is for so many of them. Lisa, as the person who's documenting this whole process, and a person who has type 1 diabetes herself, what emotionally is this experience like for you?
Yeah, no, it's a really good question. I wear two hats, right? I'm directing this film with Guy, and I'm a filmmaker first and foremost when I'm out in the field. Yet when I hear a piece of good news that, cells are reproducing, there's some good news, in the pipeline, I can't help but get, obviously very excited and inspired by what I'm listening to. But it's not normally until I'm in the car driving away from ViaCyte that I kind of allow myself to be the patient. Because when you're doing a and I don't want to say I'm like shut down automaton, like I'm not. I listen and I am just, yeah. Obviously I can't imagine anything better than not living without this disease and without, we get emails every day from people. I'm preaching to the choir here, but, there's, it certainly would liberate us all from the daily rigors of this disease and the fear of complications. But yeah, it's when I'm driving away from ViaCyte that I feel like I can breathe this, I can take a deep breath and say, wow, this is actually happening. And yeah.
So now that we're all incredibly excited and potentially a little bit verklempt, the questions about the product and the timeline for the product. Is there any timeline right now? There's this, as you have said, this is groundbreaking work that's being done right now. Paul, is there any way to estimate at this point when this product might come to market? What are the variety of steps that need to be taken before that can happen?
Sure. Yeah, as you said, it is groundbreaking. So a lot of what we're doing at this stage is what I'd call data-driven timing. Okay, so in other words, we're being driven by the data that we're revealing and we're understanding. This is the first time anybody has used a differentiated embryonic stem cell to treat diabetes. It's the first time anybody's put a cell therapy product in a device meant to protect it against the immune system into a patient. So all of these firsts are important, and we learn a tremendous amount. Interestingly, in this first trial, as I mentioned, we're in the first cohort of patients where we're giving a subtherapeutic dose. That in addition to the cells that we're giving in kind of the therapeutic device, if you will, we're putting in sentinels. And actually, I can do some show and tell. Hang on for a second here. So this, let's see if I can get this. Okay. This is actually the therapeutic device that is used. You can see it's very flexible. It's very, very thin. Okay. And it gets filled with cells, and then it's planted under the skin, in the arm, or the back, or various locations. And for the clinical trials, we do dose ranging, so we use different numbers of these to get to a different dose. So at this current stage, in the first cohort, we give a low number of these, if you will, to give a subtherapeutic dose, so we don't expect to see efficacy. However, at the same time we're doing that, we are also implanting these patients with what we call sentinel devices. And the sentinels are the devices we use in the mouse. Let's see if I can get this on camera here. So here, this one is the therapeutic device, the dose ranging device. And here is the sentinel, and you can see it's much, much smaller. This is what's called a DC20, EN20. This is an EN250 in that reflect time.
It's a little, Paul, it's a little, on the screen it's a little hard to tell the actual sizes. We can see the relative sizes of those two devices, but it's hard to tell the actual sizes. Can you like hold them up maybe next to your palm so that we can get a better sense of their sizes?
Sure. It's this one, the Sentinel device is really the workhorse for what we're doing right now, and it is about the size of your thumbnail, okay, to give you a feel for it, okay. So it's a small device. And what we do is we put 6 of these, up to 6 of these in each patient. And I got to give a shout out to the patients that are participating in this trial right now. I think they are really working hard as we are to move forward and advance this program because they're getting subtherapeutic dosing right now, but they're giving us the information we need to make this effective, or at least we hope to make this effective. So these sentinels are placed in the patient, up to 6 of them. And then what we do is we go in and we pull these sentinels back out periodically. So at like 1 week or 4 weeks or 12 weeks or whatever time frame we think makes sense, we pull one or two of those sentinels back out. And then we subject them to histology, meaning we essentially fix them and then cut them open, and we look at the cells, and we look to see, okay, are the cells surviving? Are they proliferating? Are they differentiating? Are they getting vascularized? So this is what's really allowing us to develop the techniques and the approaches that we use to make sure that these cells engraft successfully before we move to the next cohort where we give a higher dose and look for efficacy. Whoa! Sorry about that.
No worries. Oh, that's a first for me in a live interview. I've never had anyone's computer fall off.
Yeah, there you go.
Congratulations, Paul. You
ViaCyte is all about firsts. So.
Well, as long as that has caused you to pause for a moment, I have some questions about that from the chat room.
Okay.
Because you mentioned a few things. You mentioned that the number of implanted devices depends on the dose that's necessary, and folks are wondering roughly how many implanted devices. Is it something that's going to be dependent upon a person's weight or height or insulin requirements? Are the implants, is the thought that they'll be scattered around the body or in one particular spot, and where would that spot be? Can you give us some more of those kinds of specifics?
Sure. So first off, you shouldn't get too caught up in the device I showed you, because this is a dose-ranging device. So in this part of the trial, we want to be able to answer some of the questions you just asked in terms of how much dose we need to add and such. And so we need to be able to vary the dose. And it's interesting in cell therapy, with most drugs, we think of, you know, you take a pill and there's a number of 100 milligrams or 300 milligrams of that dose of whatever drug you're getting. That's not what we do in the cells. The cells actually will proliferate and fill that device. So the dose is actually dependent on the device size, because the cells will proliferate and fill the device. So the dose ranging device I showed you, we use multiple of those. Right now we're putting in 2. We think based on calculations to get fully therapeutic we may put in 6, but don't get caught up in that. Once we get to a commercial device, when we distribute this broadly, assuming we prove that this is effective, we are developing larger capacity devices where you would use 1 to 2 of these devices implanted to give you a fully therapeutic dose. In terms of, you know, different weights and such, certainly I think that it's not as big a issue as it is with some drugs, we don't believe, but we'll certainly investigate that as well as we go forward. So eventually, to summarize what I just said, eventually we expect it to be 1 or 2 devices. In terms of where it would be placed, that's one of the things we're investigating in the clinical trial. We believe as long as it's well vascularized, it can be placed almost anywhere. Just like with insulin, when you inject insulin, you can inject it in different locations. The key is that it has access to the vascular system.
Fantastic. And we have a question in the chat. We have someone who confesses to being impatient about this in the chat room, and I think, Katrina, probably most of us could confess to being impatient about this progress. But Katrina is interested in knowing if you know yet when the actual therapeutic dosing phase of the trials will happen, when you'll get past sort of the safety and efficacy part and into actually trying this out in people to see basically if it cures their diabetes.
So first off, let me say that we're very impatient. we really are excited, and you know, it took a long time to get to the stage where we're now testing in patients, and we really want to see results. And so, but at the same time, you know, we have to think about the welfare of the patients, and we have to make sure that what we're doing is safe, well tolerated, well thought out, and we have to, you know, pay the patient is number one. Actually, we have a our kind of philosophy at the company that it's on the walls, and it starts with the patient, and that's what we look at. So in terms of timing, in terms of the second cohort, the one thing I can say is we just reported a few weeks back at a meeting up in San Francisco, we reported some results. We are a for-profit company, so we have investors, and we need to raise capital. So from time to time, we go out and talk about what we're doing. And so we made a presentation at a event, and we talked about some data from the clinical trial. And it really was one of the more recent patients where we have developed these techniques. As I said, this is a data-driven process. It's an iterative process to implant, to withdraw these sentinels, see how the cells are doing, and then make changes to improve that. Okay, and then you do that again with the next patient, pull out the sentinels on 4 weeks or 12 weeks. So you can see it's kind of a long process to do this, but we learn from every patient, and hopefully we build on that learning. So as we get to the next one, we've now done 12 patients at this subtherapeutic dose. And in the latest, one of the latest patients with our latest way of treating them, if you will, we've seen some very promising results. We can't say that we're there yet, but we've seen cell survival out at 12 weeks, which is pretty impressive, okay, to get past it. If you're going to lose cells, you're going to lose them early on. So once you get out to about 12 weeks, that's what you really want to look at. And we see good cell survival in this patient. We see vascularization, so the device is getting a network of vascular vessels, blood vessels, laying across the device. And we see differentiation and proliferation. So differentiation meaning that they are becoming endocrine cells and producing insulin and showing markers of insulin production. So all of that is very encouraging and very promising. As I said, we feel there's still room to improve, and so we are still iterating from that patient. Now we took what we learned from that patient, we say, okay, great, we've made this progress, now let's look at this, and then once we get that patient implanted and pull from them, then we can compare back to that and see if we made an improvement. So far it's been kind of a steady improvement, which is great. That's what we want to see. But at some point, hopefully within this year, we will have convinced ourselves that we've maximized this and that it is good enough, and then we would move to the second cohort and start looking for efficacy.
We have a couple really interesting questions in the chat room about some of what you just said. One is about alpha cells. We always talk about beta cells when we're talking about type 1 diabetes, and we tend to neglect alpha cells, which it turns out are also pretty important. Are alpha cells going to be? Are they part of this project at all, or do these cells that you're creating from from stem cells only proliferate into beta cells?
No, actually, it's a great question, and it's really one of the reasons we chose the product that we're pursuing. As I mentioned earlier, we differentiate up to what we call a pancreatic progenitor cell, or pancreatic endoderm cell, and it's a progenitor because it continues to differentiate after we implant it. And by stopping at that progenitor stage, what happens is it continues to differentiate, and for all intents and purposes looks like an islet after it's finished differentiation, meaning that not only is beta cells present, but alpha cells and delta cells and all the various factors, glucagon and insulin and somatostatin and amylin, all of those are produced by these cells. And we do think that's a significant advantage, even though in type 1 diabetes, clearly the major defect is the autoimmune destruction of the beta cell. There's also good data out there to suggest that there's also dysfunction of the other cell types, glucagon production and such. And so we think not only is this have the potential of controlling blood glucose using insulin, but giving the fine kind of control that you really want by having all of these other agents, the counter agents like glucagon present as well.
Awesome. More questions in the chat room for you about this. First off, have there been any side effects so far, negative side effects, from any of the implants that you've done?
Yeah, so far, I'd say as we look at the trial, if I kind of ticked off what we've seen so far, the first thing we look for, of course, is safety, and that's our key concern when you're moving forward with a new product like this into a patient population. That's a big reason why you start at a low subtherapeutic dose, is because you need to understand if it's going to be safe. Obviously, we do a lot of preclinical testing to show safety, but until you get to patients, you don't have the kind of definitive results you want. So what I can report so far, only 12 patients, so put that caveat in there, is it's been safe and well tolerated. We have seen adverse events, but the vast majority of the adverse events are associated with the surgery, and that's not surprising. It is a surgical procedure where we implant these under the skin. So the product itself, though, has been safe and well tolerated, and so we're really pleased with that. Another finding, if I kind of went down the list, and it wasn't the question, but just another thing to make, is that the device that I showed you that's meant to protect these cells against the immune system, we look at that very carefully. We look for what it's really trying to prevent is any ramping up of what's called the adaptive immune system. This is the system that produces antibodies and allows you to reject organs and foreign bodies and such. And so we do a lot of testing to see if the device is actually preventing the activation of that adaptive immune system. And what I can say again, so far with 12 patients, we've seen no evidence of the adaptive immune system being activated. We haven't seen any autoimmune response to the cells that we're putting in, which is great. And we haven't seen any sensitization, which is another way of knowing that the immune system is being blocked from these cells. So far, the Encaptra drug delivery system, which is what we call this device, appears to be functioning as expected. And then, as I just reported, at least in kind of our best patient to date, we're seeing good cell survival, good proliferation, good vascularization, and differentiation to insulin-producing cells. So, I think it's making good progress as we go along.
Lisa, how much of all of this process are you able to actually document? We're talking about things that happen surgically. Are you actually able to get in and document all of this? I think we have folks in the chat room who'd be really interested to see, like, what does this process actually look like?
Yeah, no, it's a great question. So we actually have filmed one of the surgeries. We filmed a patient, the first Canadian patient. Yes, can you hear the plane? The first Canadian patient in Edmonton last August. And because ViaCyte is collaborating with Dr. James Shapiro at the University of Alberta, who was a pioneer with Dr. John Laikind of the Edmonton Protocol, which you have such an informed audience, they probably know about the Edmonton Protocol. So, here's Dr. James Shapiro, a rock star in his own right, working with ViaCyte, and we were there filming the patient undergoing the surgery, and in fact some of the footage is in our current teaser. We haven't had that much access to patients in cohort 1, but in cohort 2 we do plan on filming the patient selection process, and as well as the patient actually going through the surgery and then the follow-up appointments. I think it's, this is when they're, as Paul has said, this is when they're getting a therapeutic dose, and this is when perhaps the aha moment will happen. So if we can be there as filmmakers, I mean, how extraordinary would that be?
So as Lisa knows, the company actually has to be arm's length from the patients, so we don't have any direct interaction with the patients. The patients are enrolled at the clinical sites where we set up the protocols and such, and right now that's at the University of California, San Diego Medical School Center, and at the University of Alberta in Edmonton with Dr. Shapiro. And in case somebody out there didn't know what the Edmonton Protocol is, that's the use of cadaver islets to correct disease, and it's used in very high-risk patient populations. There's been about over 1,000 patients have received cadaver islets, and it is a very effective treatment. It is essentially a functional cure for those high-risk patients, and with current immunosuppressants, it goes out as long as 5 years or longer that patients are completely insulin independent after the treatment. So it's a good kind of confirmation that this type of approach of a cell replacement therapy is doable. But we have to stay arm's length from those patients. The sites enroll them, and so when she goes out to talk, when the film crew goes out to talk to a patient, that's arranged with the clinical sites, not with us.
Yeah, and I might just add, I mean, we obviously don't want to compromise this trial in any way, shape, or form because we have a camera there. And we don't want to ever release any information that shouldn't be released. Obviously we defer to the timeline that ViaCyte gives us and to what we can say and what we can't say. At the end of the day, I think we all want this disease cured, and I could not imagine getting in the way of that.
Absolutely. And the tone in the chat room on 2 Diabetes right now, as folks are listening to you both, is we have a number of people basically saying, sign me up, I'd like to try it now, I'm very ready now, and how deep is the incision? So there are some sort of practical questions about, what are we looking at in terms of invasiveness? And then people are just really excited about this, of course. In terms of how deep is the incision, one of the questions that actually came up a while ago was, how long do you expect? And I know that it's really too soon to ask this, but maybe there can be some guessing or what your thoughts are so far about how long the device will work. You had said that in the mouse models, you used the devices, tested them for 6 months. Was that because that was just sort of an arbitrary cutoff point, or is that the point at which the immune system started to get involved? Is there a thought that these devices would need to be removed and replaced periodically, or that it would be a permanent thing?
Okay, so let me take those in order. Okay, so in terms of the invasiveness of the surgery, it's subcutaneous. It's under the skin, so under the layer of skin. It's a fairly simple surgery. It's an outpatient procedure. Right now, we use general anesthesia, but the physicians who do it believe they could do it under local anesthesia. But at this early stage of the trial, again, we don't mess around. We want to make sure that everything is done safely. So I think it's a fairly simple outpatient procedure. So your second question was duration. Okay, that's a great question, and certainly one we're very interested in knowing the answer to. I'm not sure where the 6 months came. If I said that earlier, I'm not sure what that came from. But in the animal models, we've been out to the life of the animal, which the model we use is a mouse model, and we've been out to the life of a mouse, which is only a year. Okay, so, and at the end of that year, it's still functioning. Okay, so all we can say is that it's effective for the life of this particular strain of mouse. In terms of what we think will happen in humans, we do believe it will need to be replaced eventually. That's why when I call it a cure, I usually call it a functional cure, okay, because it's not a permanent cure. So we think that we will need to replace this. And interestingly, it's kind of a plumbing issue, if you will. If you think of this device, it has the really active part of this device is a semi-permeable membrane, meaning that it allows the free flow of oxygen and nutrients and even proteins to go back and forth across this membrane, but it isolates the cells inside. The cells can't get out of the device, and the host cells can't get in, and that's how we block that immune system. But the other thing we're blocking is the body's natural cleanup mechanisms for dead and dying cells. And so as cells turn over, you have certain cells in the body called macrophages, which come in and essentially remove that debris, okay, and clean up after that, those dead and dying cells. Well, those macrophages can't get into our device. And so there's no way to kind of clean up that debris in the device. So over time, our expectation is you'll essentially follow the membrane to the point where it will no longer function properly. So we would then replace it. Now, how long that'll be, we don't know. This clinical trial we're running right now, we will measure efficacy after we have put in a fully therapeutic dose for at least 6 months in all patients. But we will continue out to at least 2 years evaluating the patient. So that'll give us an idea on how long this might last. We're hoping that at 2 years it's still functional, and then we could potentially go even longer. But in talking to a lot of patients with type 1 diabetes and asking them the question of, well, how long would this need to last for you to be interested in this product? Invariably, what we hear is, oh my gosh, oftentimes we hear, if you can free me of this disease for a month, I'm going to be thrilled. But certainly a year or 2 years without having to worry about the disease, it would be, before it had to be replaced, would be a tolerable duration.
We have some folks in the chat room asking about eligibility, and this, again, it may be too soon to really know this, but do you foresee there being any limitations on who is eligible to use this device, or for whom this device will work, relating to age and duration of diabetes, and potentially to whether or not somebody has complications from diabetes?
Yeah. So those are good questions. I'd say right now in this initial clinical trial, there's very specific restrictions on who can go in, and it's based on age, it's based on the health of the patient, and various other things. We're looking at generally adult patients with type 1 diabetes that are C-peptide negative, that have well-controlled disease, if you will. Okay. Having said that, that's because in this first stage, we want to, we're trying to learn. We want to have it as a controlled study as possible. But as soon as, knock on wood, we show this is effective, then we would start expanding that patient population. And we believe that it could be useful in virtually any patient with type 1 diabetes. We also actually believe it could be effective for insulin-dependent type 2 diabetics as well. But we haven't studied that at this point. But theoretically, there's no reason why it wouldn't be a benefit there. The difference is, in type 1, it could be essentially a functional cure. In type 2, you'd just be a treatment, an adjunct treatment, because there's many other differences between type 1 and type 2, as you all know. So I think we're actually very interested in the high-risk patient population, both with this product, and we have a second product using these cells that is more targeting that high-risk patient population, especially patients with hypoglycemic unaware syndrome, where they're at very high risk for the complications of using insulin. So we believe that this should be broadly applicable, but time will tell. That's why we do clinical trials, and that's why we do it in a very methodical way to understand and fully appreciate and apply the product appropriately for the benefit of the patients.
We are nearing the end of the hour, but I want to make sure, here's one of the most important questions that I can ask and that we can conclude with, and several members have asked this, is how can people, regular people like myself and our members, support the work that both of you are doing, both in the creation of the functional cure and the documentation of that process, and educating people all about it. Lisa, I want to start with you, and if you could also talk about your plans for the human trial in terms of when you think you might be able to release it and where we can look for it, that would be great.
I'd like to know that one too.
Yeah. For asking that question, I'll address Paul's question too. Yeah, so we're an independent feature documentary, which means that currently we are in our fundraising mode to keep the cameras rolling. So, we believe in this power of this film to make a huge difference, to bring global awareness to diabetes, specifically type 1, on this, very powerful platform. So, if people want to support our film, if you could please go to our website, thehumantrial.com. You can buy a T-shirt. Shepherd Fairies designed it for us, and a portion of the price of $30 goes to support the film. And if people want to get more involved, there are options on our website as well. I think right now we're very keen to film a lot as Paul and his company are gearing up for the second cohort of patients. So, the time is now. And we just want to be there filming it. What was the other part of the question, Emily? I forget. Was there another part?
I think you answered it.
Okay. All right. Well, thank you.
When you think it might come out was the other part.
That was the other question, yeah. So, it is dependent on when ViaCyte wants to release the data of the trial. Ideally, we can have a release of 2017. That's our ideal release date. And we like to show it at film festivals like Sundance that are just going on right now in Park City, or Toronto, the Toronto Film Festival, where insulin was discovered at the University of Toronto. There's a wonderful tie-in there. We have so much interest in this film right now, or in general, and we know there's a global audience for it. We have people writing in from all over, and in fact, we will be following Paul to Riyadh, Saudi Arabia next month as he presents some of his findings at a stem cell conference there. So, and why are we going to Saudi Arabia? I mean, for many reasons, not just following the story, but diabetes is, there's the highest incidence of diabetes in Saudi Arabia. So this film is international in scope, just like the disease.
Absolutely, and actually, a lot of us saw the pictures of the musician Usher wearing the Human Trial T-shirt recently all over social media. That was also really exciting, just that extra bit of eyes on the issue.
Yeah,
Paul.
Amazing.
Yeah, I think for us, as I mentioned earlier, we have had really strong support from the patient community, and that has been a big benefit to us. I think JDRF, the major international group, nonprofit organization that is really focused on supporting patients with type 1 diabetes and really searching for that cure. I think their current motto is type 1 to type none. So JDRF has been strong supporters of the company, so I certainly encourage people to support JDRF. They have provided not only technical support but financial support. I think we're one of, if not the largest project that they've supported, single project that they've supported with their funding. The other one, if your listeners are in California, the California Institute for Regenerative Medicine, which was formed from the proposition that passed about 10 years ago to support embryonic stem cell research. They have provided a great deal of funding to us, again, technical support as well as financial support. They are coming up to the point where they are reaching the end of their funds, so they are going to be out looking to raise additional capital to continue CIRM. So I think supporting that would indirectly support us as well. And then just as a patient community, as we move this product forward, as we go through the regulatory process, I think as everybody knows, this is a very highly regulated space. And we work with the FDA and the Canadian health authorities and health authorities around the world. The patient community has a big and increasing influence on those regulatory authorities. And as we push this forward, I think having that patient community behind us and really standing on the sidelines and really having their voices heard on how important it is to them to see this cure, that will be a big benefit to us.
Wonderful. Thank you both so much for your time today and the incredibly important and wonderful work that you're doing. You're getting a standing ovation in our chat room, both of you. And we really appreciate your time, and we're going to keep following the work that both of you are doing.
Thank you, Emily.
Thanks, Emily.
Take great care, you guys.
Bye.